Yes, Krishna, I'm sharing the screen. Can you see the screen? I think we can start. Krishna, are you there?
Yes, I'm here. So yeah, I hope that you all watched the video. So let's move on to the next topic, the next slide. So it's about prokaryotic. So y'all know that now archaea is prokaryotic. So I'll explain y'all what is prokaryotic. So if we break this word into two, pro and karyotic. So pro means before, karyota means a nuclei. So these organisms, archaea, evolved before the true nuclei was formed. True nuclei means genetic material that is membrane bound. So archaea is derived from the Greek word archaeos, meaning ancient or primitive. So until 1960, the scientists believed that archaea is kind of bacteria because archaea is prokaryotic, same like bacteria. And also they're exclusively singular cell organisms. But they found it's not true. Next slide. Yes. So here you can see the polygenetic tree. So bacteria is taking the branch from the common ancestor separately, while you can see archaea and eukarya are sharing one common branch, and then they are dividing into two. So from this genetic studies, scientists found that bacteria is prokaryotic and also similarly archaea too, but archaea is sharing much similarities with eukarya than bacteria. So that's why they have categorized this way. That means eukarya and archaea, they are sharing many similarities than archaea with bacteria. But archaea and bacteria, they are prokaryotic. So next. Yes, though archaea are prokaryotic, cell walls are different than of bacteria. Archaea closely represent eukarya. I hope you all understood what was represented by that polygenetic tree. So I encourage the students to take notes while I explain, so that will help you to understand more. So next slide. Yes, so here is the basic comparison. So yes, here we have archaea and bacteria. So we are going to compare what are the differences. So composition of cell wall. If we look at the composition of cell wall, archaea and bacteria, archaea has peptidoglycan. So while bacteria, they have peptidoglycan cell wall. And also when it comes to cell membrane, their cell membranes are lipid monolayer, mostly lipid monolayer. But when it comes to bacteria, they have bilayer and membrane lipids. So when we consider about the membrane lipids there, archaea, they have ester-linked, while the bacteria have ester-linked lipids. And also archaea, they have main tRNA called methionine, while bacteria, they have formyl methionine. And when we consider about the number of DNA polymerase, archaea shares multiple DNA polymerase, while bacteria shares single DNA polymerase. These are the basic basis of comparisons that we can study. So you can see actually though the archaea and bacteria are prokaryotic, these things are different for them. Next slide, please. Yes. So when we consider about the types, usually when we name these archaea, with these names itself, we can identify halophiles, thermoacidophiles. Halophiles means they prefer more salt. Philes means loving or preferring. So when you see such words, halophiles, thermoacidophiles, you know those are archaea, not bacteria. So here thermoacidophiles means they prefer temperature and acidic environment. So thermoacidophiles. So when it comes to bacteria, we can have main two types, gram positive and gram negative. So let's look at what's pathogenicity. So when we consider about archaea, they are non-pathogenic, while bacteria can be pathogenic or non-pathogenic. They can cause illnesses or sometimes not, because there are some bacteria, they benefit us as well. So when we compare the living environment, archaea, they prefer very extreme conditions such as volcanoes, hot springs, salt marshes. So they are very unique. They occupy very unique, harsh conditions to live. So when it comes to bacteria, they live in all ecosystem. It occupies a range of ecosystem. So, and also when we come to the antibiotic sensitivity of archaea, they don't show any sensitivity towards antibiotics, while bacteria shows sensitivity. For examples, if we see the examples here, we have listed few, so you can refer them later. So next slide, please. Yes, here also we have a video to watch. We will be sharing that as an additional reading material. So actually these archaea, similar to bacteria, they undergo binary fission. So I'll be explaining the same thing in bacteria, so I can explain there as well. So we can send this video. Yes. So yeah, I'll be explaining the same thing in bacteria, don't worry. So here there are some examples we will be discussing now. So there are five main archaea. Yes, archaea. So next slide, please. Yes, our first example is Thermococcus littoralis. So these archaea, they are according to the shape, we say coccus means they are spherical in shape. So these they prefer very high temperature environment. So they are found around deep sea hydrothermal vents. So don't worry about this. I'll be explaining this hydrothermal, what are vents. And when it comes to its size, it ranges from 0.5 to 3 micrometers in diameter. And also, this Thermococcus littoralis is non-motile and they prefer slightly acidic water. Next. I hope everyone are taking their own notes. So I again encourage you all to take your own notes. So our next example is Pyrolobus femiri. So this archaea, bacteria got the ability to live at extremely high temperature. They show a very significant feature, because when the temperature is low, they can freeze. they can solidify their body according to the temperature change. So that's significant to this Pyrolobus femiri, and they also found in hydrothermal vent systems. Next. Our next example is Nanoarchaeum ecotens. So this species also found in hydrothermal vents, and they are thermophile. They like high temperature environment. And here the importance is this Nanoarchaeum, they share, like they are an obligate symbionts on Archaea, like Archaea ignicoccus. Ignicoccus is another archaea. So these Nanoarchaea, they are in symbiotic relationship with Ignicoccus, and they have circular chromosome. Next. Yes, our next example is Halochondrata. So this is something very specific. They can do photosynthesis, and rare shape, their shape is square, flat box-like, and these archaea, they got vacuoles float in water, and it helps to capture sunlight to and they can make a sheet-like thing joining us. Next. Yes, our last example is Ignicoccus. So I said already, this Ignicoccus forms a symbiotic relationship with Nanoarchaea. So these Ignicoccus are, they also live in hydrothermal vents, ignicoccoid cells, very small ones. They exhibit a smooth. So I hope you all got a good idea about the specific about the Archaeobacteria. So next we have a quiz prepared for y'all. So we will be displaying the questions. So you can send your answers typed in the chat. They are very easy questions. I hope y'all took notes, and yeah, you can answer these questions. Your answers typed So I hope students are answering the questions. Yeah, so you can check the answers with us. So keep posting your answers. So what's the cell composition of bacteria? We have asked. So when it comes to the cell composition, cell composition of archaea is pseudopeptidoglycan. Bacteria, they have peptidoglycan cell wall, while archaea have pseudopeptidoglycan. Yes, that's the answer. So do bacteria have sensitivity for antibiotics? Yes, they do. So the next is state one fact about Helicodratum. So I've shown a picture here. So you can see, I said that they have an unusual shape of cell. So they are square in shape, and also they got gas vacuoles, so which help them to float in water column. So that way they can capture sunlight and produce their own food. So what type of reproduction method is exhibited by archaea? Yes, it's binary fission. So one cell divide into two similar cells. So I'll be explaining about binary fission in coming slides, don't worry. So which species solidifies under low temperature? So I showed you, when I was explaining, I showed the example. So Nanoarchaeum equitans is the species that solidifies under low temperature. So we can move to the next slide. Next. So here is the reflection activity. So we'll be giving this in the end of this session. So yes, we can move to bacteria. I hope that y'all got a good understanding about archaea up to now. So we will be discussing about bacteria from here onwards. So the learning outcomes. So from this learning this bacteria session, so you will be able to list the features of bacteria and major differences between bacteria and archaea. So we will be again refreshing the knowledge with that chart. So able to describe the importance of bacteria in marine ecosystem, name few examples. So next slide, please. Yes. So when it comes to bacteria, you know that like archaea, similarly, bacteria are prokaryotic and they are microorganisms, which means you can't see them through your naked eye, but through microscope, with the aid of microscope, you can see them. So typically they are few micrometers in length, very small. And bacteria have a number of shapes ranging from spheres to rods and spirals. So we will be learning about them in the coming slides. So this is the structure of a bacteria cell. So here you can see they have mainly cell wall, cell membrane, and they have a membrane, and fifth bacteria, they have capsules. And also there you can see small elongations, extensions from the cell of this bacteria body. So we call them pili. And one which is very long, we call it flagellum, which helps bacteria in their locomotion. So those are the main things that we want to discuss here. Next slide. Yes. So according to bacteria shapes, their shapes, we give different names. So here you can see spherical ones, we call them cocci. So when they are round in shape, we call them cocci. So when two round cells together, we call them diplococci. And when we have a chain of this spherical bacteria, we call them streptococci. Again, when it's a kind of group, we call it staphylococci. And when 4 cells are together, we call them tetrad, and 8 cells together, sarcina. And when it comes to rod also, we can have chains and one single cell with many flagella, and then spirals, one curved bacteria with one flagella, and then one with in a point where many flagella extend, like with one point many flagella they come out. So, and also we have some wavy coil shaped one, we call them spirochetes. So these are according to their shapes, we can name them this way. So next slide please. Yes. Bacteria inhabit soil, water, acidic hot springs, radioactive waste, and deep portion of Earth's crust. So actually, bacteria, they can live in all ecosystem, and also they form symbiotic as well as parasitic relationship with plants and animals. So let's see what's symbiotic. So symbiotic means is any type of a close and long-term biological interaction between two different biological organisms. So simply we can say living together. Two organisms, they are living together. So there are types we can, according to their interaction, we can divide them into 3 main types. One is mutualism. Mutualism means when two biological organisms live together, they benefit each other. So a relationship in which both species benefit. When it comes to commensalism, is a relationship between two species in which one benefits and the other is unaffected. So that's called commensalism. So when it comes to parasitic relationship, in which one species benefit and the other suffers. So other is like the one organism that get harmed. So that's called parasitic relationship. So the next. Yes, here I have showed a hydrothermal vent system. So this kind of ecosystem, or we can systems, are found in deep oceans. So hydrothermal vents are cracks in the earth crust where geothermally heated water leaks out. So when we consider the ocean floor, there are some cracks in ocean floor. So through that cracks, water seeps in, and we know inside the earth crust it's very hot due to magma and the depth. So when water reaches this heat, they get heated, and then same time they leach other minerals as well. So once these hot water finds a way out, pressurized way out again to the ocean water, they come through these hydrothermal vent systems. So once they meet the cold ocean water, this boiling, very heated water, when they meet this cold water, they start to precipitate and form chimney-like structures shown here. So hydrothermal vent is a fissure in the earth surface from which geothermally heated water comes out. So you all can see. So the significant here is, so we have a kind of bacteria which is unique to this ecosystem. So as it's very in dark environment where no light penetrates, so the bacteria live here. They use the inorganic substrate to form their food, and also we can call them chemosynthetic bacteria. So they form the basis of the food web in such ecosystem. So they are not photosynthesis. They don't use light. It's very dark in this ecosystem. So they use inorganic substances that comes out from this hydrothermal vent, and it's very hot. Okay, so these chemosynthetic bacteria grow into thick mat-like covering. So these chimneys are covered with this mat-like chemosynthetic bacteria. So in the next, you can see in the red color, it's covered, like those are tube worms living near the hydrothermal vent. So these kind of organisms like crabs, snails, tube worms, and fish, they feed on this thick mat of bacteria. So that's the significance there. So these thermal vents are, we can say they are biological hotspot, because the organisms that found in this ecosystem we cannot see in any other ecosystem on planet. So next slide, please. Yes, here you can see how the crabs near hydrothermal vent, they have covered the chimneys. So this is how the crabs live in such ecosystem, hydrothermal vents. So this they are very white in color. They lack color, and also most of the animals, shrimps, they don't have eyes. So that's the uniqueness of this ecosystem. Yes, next slide, please. Yes, here I have pictured bobtail squid and Elevibrio fischeri. So this is to show a symbiotic relationship. So bobtail squid, they also live in the dark, deep ocean. So they harbor this Elevibrio fischeri bacteria in their mantle, so they can generate light. So that's the specialty. So in return, yes, they receive sugar and amino acids in exchange for light. These bacteria, they get sugars and amino acids from this bobtail squid while they generate light for the squid. So they are in a mutualism. So they help each other. So I hope that you got an example for this symbiotic relationship with this bacteria. So next slide. Yes. Yeah, we have here another bacteria example. This is called sulfur pearl of Namibia. So the largest known species of bacteria was found in 1999 in the ocean sediment of Namibia. So here you can see inside the cell small yellowish spots. So those are element of sulfur element, so which they have taken inside their body from the environment. So the shiny appearance and the tendency of cells to group together in a long string. So that's how they got the name with this yellow spots called sulfur pearl of Namibia. So next. Yes, so here we are focusing about the reproduction. So about reproduction of bacteria. So binary fission. So that's asexually how they reproduce. So they have binary fission. So where a single cell can split into two identical cells. So this is how archaea also reproduce. So binary fission in prokaryotes, which is applicable to archaea as well. So here you have asexual reproduction method for bacteria is binary fission. Next. Yes, and also we should know that bacteria, they can sexually reproduce. So we call that method bacterial conjugation. So I showed you in the picture, we had a small extension from the cells called pili. So through that, bacteria, they can contact the next. So there are two types of bacteria cells. One, we can have F plus, F and positive cells, and F negative cells, which is donor and a recipient. So here, this F positive cells, what they do is they connect through pili and they exchange one strand of DNA to the other, which has not got one. So that way, both can, in the end of this process, both become F positive bacteria. So this is how the sexual reproduction happen in bacteria. So the next slide. Yes. So when it comes to the importance of bacteria, so they are the main decomposers in marine. So we can have other decomposers as well, but they play a major role in decomposing the waste. So here you can see there are phytoplanktons, zooplanktons, as well as other marine organisms. So once they die, they settle into the ocean bottom and they become waste. So bacteria, where they come into action and they decompose this waste and they produce nutrients. They release the nutrients that was already deposited in the living organism's body. So mostly we can say in tropical oceans we have plenty of these decomposers bacteria than the Atlantic or Arctic oceans. So that's very important. And also without this bacteria, it's very hard to do cycling of these nutrients. So there will be a dump of waste without being decomposed. So there will be many issues. So they play a very good, very important role in decomposing this waste. And also we can have other decomposers I earlier mentioned about, like fungi, any other marine organisms like echinoderms, crustacean, mollusks. They also contribute in decomposing this waste, but they are macro decomposers. We call them macro decomposers. These are micro decomposers. So next slide. Yes. So carbon, nitrogen, sulfur cycles are supported by these organisms. The movement of this from the living matter, once it comes to the dead material or waste, they release the nutrients out. So we call it cycling. And carbon and nitrogen cycles are two of the most important global cycle, and bacteria play a key role every step of the way. So we will be studying that in our next slides. So next slide, please. Yes, this is about carbon cycle. So I'll be explaining this cycle, 3 cycles. So I hope that you take notes, you can draw this picture, and then later you can describe. So here, mainly we have carbon dioxide in the atmosphere. So where there's carbon dioxide in the atmosphere come into interaction with this marine water. So this is representing only the biological influence. So this carbon dioxide in the atmosphere uh, can be fixed by these phytoplanktons. So phytoplanktons, they can be consumed by the zooplanktons. So, and then other marine organisms like fish also, they can feed on this phytoplanktons, and they can fix this inorganic CO2, carbon dioxide, into organic carbon. So once they die, they settle in the ocean bottom. So there comes the bacteria into play. So what they do is they secrete extracellular enzyme, and they help degradation, or they decompose. So that way nutrients and smaller organic matters are released to the ocean water. So the same small organic matters, they can be uptaken by other organisms as well as these bacteria. So the most of these organic released carbon, they can be settled in the ocean bottom, so they can be converted into long-term storage. So this is very important. So how this atmospheric carbon is transferred to ocean sediments. So next is our nitrogen cycle. So we know that most are like approximately 78% of this atmosphere consists with nitrogen. So they can be fixed by the help of microphytobenthos and some archaea. So here there is a fixed nitrogen. They can be used by uptaken by these phytoplanktons, bacteria, and archaea. That way they contribute for this organic nitrogen. And once they die, they can be again releasing. They can release ammonia into ammonia into the environment. So that way again they can end up in the atmosphere. Hello. Next. Yes, next is the phosphorus cycle. So here no major contribution is from atmosphere. So only this deals with water and then sediment. So from sediment, it's deposited as inorganic form. So once it leaches into the water, they can be used by phytoplanktons, zooplanktons, and once they die, they deposit as organic phosphorus. So then bacteria comes into play. So here you can see down. So bacteria activity. So when bacteria they do their decomposition, so they release phosphate. So that way again they are converted to inorganic form. So and also these organisms, phytoplanktons and other marine organisms, as they excrete, that way also phosphorus can be uh, added to the ocean. So that's where like from organic with bacterial activity, organic phosphorus is converted into phosphate. So that's important there. Next. So next we have two more cycles to cover. So sulfur cycle and iron cycle, that's related to ocean. So we'll be sending you some additional reading material in the group, so you can read and write about this sulfur cycle and iron cycle with pictures. We will be sending the notes so you can read and understand and make your own, you can draw your own cycle. So for all these carbon cycle and nitrogen cycle, I just showed you the picture, so you can, looking at the picture, try to understand and write your own description about every cycle. So that's what we expect from y'all. So next. Yes, this is about cyanobacteria. So the significance, can students say what's the significant in this bacteria? Do you know? So if you know, you can put it in the chat. So I can give 1 minute. So what's the importance of this bacteria, cyanobacteria? So I can give 1 minute. Yeah. Yeah, I can see they can do photosynthesis. Yes. Yeah. Thank you for your participation. Yes. These cyanobacteria, they can use light as they process the pigments to do photosynthesis. So that's something significant with this bacteria. So next slide. Yes. Yeah. Here, this is something very important. So I have put a picture here. So stromatolites. So these are ancient structures, we can say. So here in this sedimentary structures like yeah, conical, or we can say domal structures. Inside this sediment, there are cyanobacteria layers. So they form, like, they are very ancient forms, fossils, we can say very rare in today as well. So and this is believed that they released they started to release oxygen in ancient time, where the anoxic ocean started to become oxic. So they are main contributors. So these structures we can see still, some, but very rare. So these are stromatolites. So they consist biofilm layers of cyanobacteria. So how that there it comes, the cyanobacteria incorporated into these structures. It's important to know when it comes to marine ecosystems and marine related bacteria. So that's all. I think next slide. Yes, I hope that we have come to the end of this session. So thank you for your participation. So we hope to send your extra reading materials and videos to watch and understand. So we covered 1 hour session here. So you all have to work more on this. So I hope that students will be start doing their book work. So that's your output we want. So you can start working with your books, nice diagrams, pictures, and notes. You'll be taking your own notes. So we want to see the end results of this. So hope to see you all in the next